So today we're going to talk about biosensors. And when we talk about biosensors, we've got a whole range of biosensors based on different kind of physical principles that are used to detect things. So biosensors, basically, you've got something attached onto a surface, and that's going to be called the ligand. And then you're going to flow across another molecule of interest. It's going to bind. I'm going to call that the analyte. There's different types of biosensors because they're all based on different kind of physical principles to measure this interaction. Of course, you've all used biosensors already. You're probably being adept at using them because in the last couple of years, you may have used one of these, which lateral flow, and it is a biosensor. So what have you got? You've got this kind of layer, and you've got on that layer then you've got two strips, C and T. You've got antibodies against either the antibody, an antibody itself that's going to bind to the antigen, which in case was the COVID capsid protein, and a control, and then a kind of test line, which is then going to have an antibody against the COVID capsid. And of course, so what the analyte is, of course, is your solution from your nose and your throat. And that's going to mix with an antibody, which is going to bind onto the capsid protein, and that's labeled with colloidal gold, which is this kind of red color. So when you do it, it kind of goes up. And of course, if the antibody's there, it should bind then to the control line. And then if you've got COVID, then that's going to be bound this T line here, and that's going to concentrate that red line. So why do we use biosensors? Why are they so useful? Well, you can do a whole range of experiments. You might just be wanting to know, does A and B bind together? Do the thing that I get. That's a kind of a simple kind of experiment. You might want to know how tightly it kind of binds. And then you might want to do mutational analysis. So say you've got a structure, and then you want to say, I think these residues are important. Let's mutate that and see how the binding is affected. You might want to go down even deeper and say, well, what's the on and the off rates? Can I understand the dynamics of the system? And the reason for understanding the dynamics of the system is, the cell's really complicated. It's very busy. There's lots of things happening. And if we just think of, say, two things coming together, to form this complex A and B, then it depends upon not only this intrinsic association rate, K, but also then depends upon how fast this falls apart, and that's this rate D. And because it's a biomolecular interaction, then the observed rate constants then are dependent on the concentration of each of the things. So the more you have, then the faster the observed rate is going to be. But the off rate doesn't really matter what the concentration is, things will just fall apart. And then from these individual rate constants, then you can work out what the overall binding constant is. And it's really important to then think about that the level of binding you're going to get, how much complex you get formed, actually is not just related to the affinity, but actually related to these individual little rates. So the same affinity could be then broken down in different on and off rates, and it could give rise to different mechanisms, which we'll see later on in the talk. Okay, so there's just so many companies with so many different types of biosensors, and it would be exhausting to kind of go through all the different kind of technologies that they've got. So we're just going to concentrate on the ones we've got here in the LMB. And this is the dynamic biosensor switch sense, the Sartorius octet, and the Sartorius biocores. So what's a biocore? Well, this is our T200, and what it's got is the biosensor is actually this gold layer that's laid on glass. It's very, very small. And then attached to this is this dextran sugar layer, which we're going to attach proteins onto, or ligands of choice. This is then in a little plastic cassette. You see a little gold square there. That gets inserted into the machine. Microfluidics gets clamped on top to give us, in our case, then 4 flow channels. And then at the surface, we're going to buffer through one side, the dextran side, where the molecule of interest is then attached. And on the back side, we're going to shine light through a prism. The octet uses this biolayer interferometry. We're going to call it BLI because interferometry is a real mouthful to say. So it's got these little tips. It's got an optical layer, which is biocompatible. And so you attach things onto the surface here. This little tip, light gets shone through that tip. These are all mounted in these 96 well racks, which you can take rows of 8 or rows of 16. and it goes into the instrument, and then the tips then go into the microtiter plate here, and then swirls around, the plate swirls around. And this works by just incident light going on, and you get an interference pattern, as we'll see in a second. Now, the switch sense is a different kind of instrument. It's called a DNA nanolevers. So the chip here on this DRX2 that we've got, it's got 4 channels, and in each of these channels here, you've got these 4 electrodes, or so you've got 6 electrodes. And if we zoom in here, 6 electrodes in one channel, you can use one channel at a time. And in each of these spots, then there's these DNA nanolevers, which are, there's one strand attached, which has a fluorophore on it, and then there's a complementary strand, which you can then functionalize. Right, so what kind of signals do we then get? So in SPR, you can see when I'm going to talk about SPR, I have this little symbol at the top. So what we've got is we've got polarized light coming down onto the surface here. And that's going to be then reflected back off. Now and then at the surface of this gold layer, it's a very, very thin layer, and it's got surface electrons. So that when then the energy of the incident light at a certain angle matches the momentum of those electrons on the surface, then light gets absorbed. So you get resonance. This is why it's called resonance. So then if you look at the light coming back off again at a certain angle, you can see at the bottom here, then there's a dip in the light intensity, and that's called the SPR angle. And now that will depend not only on, that will depend on what the refractive index is between the solution and the gold and the glass. And so if anything binds onto that surface, then you'll get a change in the angle. And then when you start to flow buffer off, then the angle will go off again. Right? So you can look at association and dissociation. With the BLI, what you've got is you're going to have light shone on, and it's going to be then reflected back from the biocompatible surface, and also then from this control surface as well. And then they're going to interfere with each other, and you get this interference pattern here. So, and because it's white light. Now, if anything binds onto the surface down at the bottom here, you're going to get a change in how the light is then reflected back. You get a change in the interference pattern, and if we overlay the two things together, then there's a change in the maximum and minimum where the interference pattern is. And so that is then, in terms of wavelength change, it's nanometers. Now, instead of, as I said, instead of flowing things through the channel, as in back or in octet, then you have a plate, and then you move things differently through different plates. You have to set out a micro plate and put the sensors in one after the other. And the volumes you can use is for 96 well plate, 200 microliters, 100 up to 130 or down to 80 for a 384 well plate. And also an advantage in this is you can use very, very small volumes in a tilted well, 384 well plate, which can get down to about 40 microliters. Okay, so for switch sense, it's a bit different. Again, you've got these nanolevers. So you've got essentially a camera focusing onto the spot, and then there's electrodes across in the actual spot. And so they're going to then alternate the current. So when it's positive, the nanolever is then attracted to the gold surface. That quenches the fluorescence. Then you reverse the charge. Then the nanolever starts to go float up, and then the fluorescence increases. So you're getting then this alternating current, and you can see then the change in the fluorescence in the microsecond time frame. And here's kind of a movie of what's going to actually happen. So you can imagine here's the nanolevers flowing in buffer, and okay, so the fixed nanolever is attached to the 5' end. There's a 3' fluorophore, and then the complementary strand is then got a 5' free. So if anything binds, that's going to slow that motion down again. And then hopefully at some point, then your protein of interest is just going to fly off, and then the motion is going to come back to the same speed. So what we're then essentially looking at in the bottom here is we're just looking in this microsecond time frame of the dynamics of this response, of how this nanolever then changes in this alternating field. And you can do several different kinds of experiments. You can actually look at, say, how this nanolever moves, or you could have the nanolever actually just standing straight up. just to have it as a very positive charge. And then you can look at anything binding by this fluorescence, then proximity event. If you remember back, if anybody came to the fluorescence lecture, if not, go back and have a look at that when it's on YouTube. That if something binds near the fluorophore, you can change the environment, and so then you'll see a change in the fluorescence. Another thing you can do as well is that if, say, you don't have the complete nanolever and you put on a nuclease or a polymerase, then you can then start to form or degrade the nanolever, and that will change the distance of the fluorophore from the surface and get a change in fluorescence. Okay, so all these kind of different techniques, you're going to see these kind of curves. So you're going to get an association curve and then a dissociation curve. The units for SPR is called resonance units, and that relates to the angle, and that then relates also to how much mass is actually on that surface. For BLI, then it's just nanometers. It's not distance, it's not kind of size, it's just this wavelength change. And switch sense, you can have it either fluorescence change or the dynamics of switching. So which one do you choose? So many different ones to choose. Well, they've all got really, really good kind of dynamic range. I mean, I think the BLI is not really sensitive for very small molecules. They've got a good range of affinities they can actually measure. And I think one advantage of, say, the switch sense is that it can accurately measure the very, very, very slow flow rates or dissociation rates. You can go for a very long time. They can both be done in semi-high throughput. And another thing I won't talk today about is switch sense, which you can actually do some sizing as well. Right, so what are the issues? It all looks very nice, all very shiny instruments. But of course, cost can be an issue, which one you choose. Chips can be quite expensive. If the experiments doesn't work, then you have to get a new chip. The sensors, you get 96. So you can actually then do multiple experiments. But if you're only doing one experiment, then that's going to be a cost. Obviously, the fact that you're actually attaching one thing to the surface, that's going to change how the interaction happens. Or you're then going to get steric hindrance. Obviously, you've changed the rotational entropy of one of the thing that's bound. So the surface can also then have non-specific interactions, as we'll see later. And so you have to be very, very careful with controls. Buffers, you have to be very careful because Biocor is really sensitive to refractive index changes. You have to be very careful, especially with glycerol. Switch sense, you're switching a DNA, so you have to be very careful with the salt concentration. Okay, the rate limits, obviously there's response times, so if very, very fast rates sometimes can't be measured, and off rates, sometimes you can't go as long as you want to go. With the octet, you'll get evaporation. With the Biocor, then the pump is only a certain size. Switch sense, because it's got a peristaltic pump, can go much longer. And with everything, as we'll come on to later, some high affinity interactions where you've got a very, very slow dissociation will be very, very tricky to fit, but we'll see ways around that. Okay, so let's think about what we have to think about when we do experiments. This is kind of a typical workflow. And just make sure all your buffers and everything is nice. Kind of what the concentrations are. You want to avoid having things in tris buffers if you're going to couple with amides. Do you need detergent? Have you got DMSO in your system? And for SPR, it's really critical to then match the running buffer that you've got and what sample you're injecting into has actually got. So make sure your proteins are nice. You know what the concentrations are. So which way do you do? You've got something on the surface, something flowing across. Which one do you choose? Often it's just a matter of economy. You can't make one or one partner, or it could be that one aggregates at high concentration, so you can't really get up to that concentration. Or choose where, say, you're attaching something onto the surface. It's multivalent. Okay, so how much do you put on? Well, for kinetics, you want to put on for as low as you can kind of get away with for a good signal. And for equilibrium experiments, you want to have as much to get, again, a good response. So you can go through from high to very low. Now, what's the kind of rule of thumb is this. So the amount of ligand you put on depends upon what you want to get in terms of the maximum response relative to the mass of the ligand over the analyte. So you can imagine if you have, a very small analyte, say like a small molecule, what you might 500 Daltons, something like that, and you want to bind that to say a 200 kilodalton protein, then you're going to need a very a lot to actually put on the surface to get a good response. Consequently, the other way around, if you've got a small protein and binding onto a complex, then you don't really need very much of the small protein to get a good signal. So for BLI, you don't really have to kind of guess. You could just actually run a trial experiment. You can do things in plates. Everything's done in parallel. So you can put your ligand in one column at a range of concentrations. Put your analyte in another column where you hope it's going to bind very well, maybe 10 times above KD or whatever you can kind of get. And then look at the responses you get. Look at the fits, the data model, and do you get a really good fit? Is you kind of your R squared above? With switch sense, well, I mean, because you've got a set amount of DNA nanolevers on the surface, then it's really, it's 100 to 500 of DNA coupled ligand. If you've got some avidity, you can reduce the density by electrical desorption to kind of get kind of much less lower density. But it's important to remember that the actual density of these nanolevers is very, very low compared to Biocore to kind of start off with. So how do we get things on? Well, there's two kind of ways. You can either capture or you can then cross-link. So when you capture, then it'll be usually the good thing about that. You've got these different kind of antibodies or GST, and you can capture the molecule, bring in the analyte, and then you can then regenerate the entire system, capture the ligand again, and then flow it across the analyte. This works for most things. For obviously streptavidin, because streptavidin biotin, it binds, it's not going to come off, and there's nothing you can get to wash that off. So with coupling methods, then you're going to directly cross-link your ligand onto the surface with different cross-linkers. So the advantages of that, of course, is that you've got it on there, and it then is stuck. And as long as your protein is good for the length of the experiments, you can do lots and lots of experiments, take it off, put it in the fridge, come back, run it again. So there's a whole kind of plethora of different kind of chips you can kind of get. Don't worry too much about it, I mean, in terms of the actual details, but different levels of dextran. You can have specific ones for capturing different tags, and there's also antibody specific as well. And similarly with the BLI as well, you've got the same kind of flavor. You can cross-link, you've got different kind of captures and lots of antibody ones. And you can imagine that actually in pharma and biopharma, these kind of techniques are really key because to actually measure the validity of how good you've got an antibody and how that compares to other antibodies. And so there's lots of methods for doing that kind of work. In the switch sense, we have just a multi-purpose chip with either 48 or 96 base pairs of these nanolevers. You can look at different measurements. You can also look at enzyme reactions. So we've got one of the original machines, and recently the company's then brought out a new instrument which is much more adaptable, and you can actually have DNA origami surfaces, and also you can then have these Y structures of nanolevers, so you can actually then start to look at say fretting and things like protax. Okay, so how do you get and switch sense? How do you get things on? Well, if you've got a DNA substrate, if you want to like DNA or DNA RNA, then you just need to make an oligo and then flow it across. If you've got a protein, then you need to attach the oligo onto the protein, and that strand is going to be complementary to the nanolevers on the surface. So you just flow them across. And how you cross-link them, again, the normal kind of amine or thiol, or alternatively, there's different tags which have already attached to the oligo for doing these captures. So for amine cross-linking, you've got a cross-link, It goes onto the surface. Now you've got these carboxylate groups then on the surface because it's dextran. You then attach on the crosslinker, and then that becomes reactive then against primary amines here. So it'll be a bit random. If you've got a cysteine on the surface, then you can use this, a PDA, to then attach then free files here. Or alternatively, you can, and this is going to be reducible, or alternatively, you can just do maleimide cross-linking, which will permanently link things on. Okay, so we've got things coupled. How do we couple things on BioCore? Well, we can use very, very small amounts of protein going on, and we can take advantage of the fact that, okay, this surface is slightly negatively charged. If you get your protein below its pi, you put it in the pH below its pi, then what's going to happen? It's going to be positively charged. So that's going to be attracted to the surface just by electrostatics. And then that's going to increase the chances you're going to cross-link across. There's a little scouting program here that you do. And again, it's just different pHs. And you can just want to look for kind of linear kind of traces where you can then start to go, right, I know how much is going on for a certain time. So if I want to get to a certain level of binding, then if I just do it for like 2 minutes at this pH, then hopefully I'll get that. And here's an example, a theoretical example here, where you're getting a cross-link on first, you're putting then the protein at a low pH in, it's been attracted on in the middle there, then you switch into buffer, it gets washed off, except for the stuff that's covalently attached, and then you block the entire reaction then with phenol-amine. So that's benches any free cross-linker. Okay, sensors like nickel NTA, well, that's essentially just like running a normal kind of NTA column for histidine proteins. You have to put nickel on, capture the protein, and go to binding of the analyte. Streptavidin, just flow across your biotin protein, and it just sticks on. So for specialist things, if you've got membranes, they used to make a hydrophobic chip, but I think that's going out of service. So now if you're looking at membranes and you need to like make vesicles, and you can, on this L1 chip, this will actually capture vesicles, and then you can then look at things binding onto the surface of the vesicles or binding onto proteins that are actually embedded in the vesicles. Okay, so we're going to then kind of, we've got capture, we're going to do some controls to make sure we've got non-specific binding, and we might have to then regenerate. So an important thing in all these kind of experiments is having a control. So in the BioCore, you've got 4 channels. You can run these singly, or you can run these in pairs. So either 1 to 2, or 3 to 4, or 1, 2, 3, and 4. So you can use the first channel in the series as a reference, and then take that away from the signal on, say, channel 2, where you've got your protein of interest bound on. So we usually treat that exactly as we treat the channel with the ligand on there. So, you know, you might immobilize a non-interacting kind of partner or your biomolecule, or like a mutant protein that doesn't bind, or a random bit of DNA. And the reason for that is you just want to take away any kind of bulk changes. So down at the bottom, if you see in FC1, you're getting this kind of just hump going up, and that's just buffer change. And channel 2 is where the ligand is, the analyte, and then you're getting an interaction. So you take 2 away from 1, so 1 away from 2, and then you get your results here. So here's an example of a capture. So red, on this chip, we've got an anti-GST antibody. So we're going to be looking at fusion proteins, GST fusion proteins. So first of all, we flow on the left-hand side there in red. We're just going to flow on GST, just on that channel 1. Then we stop that flow. We've just then flow into channel 2, put our GST fusion protein on, let that on, that gets captured. Then we flow on the analyte over both channels. So then we're getting responses there from both. And so we then take away the reference. Okay, so it's 2, that's 1 away from 2. We also then have put in a 0 buffer, just a buffer going through. And we always do that because there might just be some instrument noise when the pistons kind of move, or just some drift in the signal sometimes. And so we put that in to then get our lovely data, and then we fit that. So you can see here right at the end on the left-hand side here, I put in regeneration solution. So if you have a look at the curves in green there, you can see, well, okay, not everything seems to be coming off, at the end. So if we have a look here at the highest concentration, it's not gone down to baseline. So what we used then in this case was, it was a low pH, and that just stripped the GST completely off the antibody. So then you have to, every cycle, you'd have to then put on more GST or more GST fusion. And so that's called regeneration. And so what you hope to happen is that when you regenerate, you get down to baseline again, and then you inject in your ligand, so your analyte again, and you get a good response. Obviously, if you don't then regenerate that surface down to baseline, you're going to get a poor response. The worst case scenario here is that you put something in, regenerate the surface, and then the second time, oh God, you don't get this, actually get any response as well. That means you've kind of destroyed the surface. So, and the reason for that is the kind of regeneration things that we kind of use are high salt or acid or alkali or detergent. So, it could be that just by trial, you'll find that, well, actually, I can't use alkali. Right. You can actually solve this problem if you can't get things off, and sometimes things you don't want to regenerate, or things just are going to just take far, far too long. You can use single cycle kinetics in the biocore. So on the left there, you can see these multi-cycle kinetics. So we're doing different cycles, starting from the start, putting in increasing concentrations, and letting things dissociate after each cycle. On the right here is a single cycle. So we're just injecting increasing concentrations of our analyte, and then until the end, where we then let everything dissociate down and fit that entire curve. And at the bottom there, doesn't really matter which way we do it, we end up with similar binding curves. Right, so let's think about one issue with biocore as well, is this thing called mass transport. Okay, so you've got things flowing in the bulk, but there's this dextran layer, and you've got to get the analyte then through the bulk, through the dextran layer, to find then the ligand on the surface there. So this is called the transport effect. And if you had a very, very high level of ligand on there, what you can get is you can get analyte depletion. So you imagine it's flowing through the channel, and then lots of it is just being bound at the start. And so the concentration, as you go down the channel, is getting less and less and less. The other thing, because what happens is basically you don't have enough flow to then replenish things that are being bound on. Conversely, then, if you dissociate off and you've got a very, very high level of ligand, it could be that your analyte comes off, but then it just bumps into another ligand and then binds again. So the dissociation then looks much slower than it should be. Okay, so you can actually fit this, but the ideal thing is not to do that, and to try to just use a low amount of ligand, and high flow rate. So here you can see on the left there, this is something where it's different flow rates, and you can see there's a different kind of interactions. So that's definitely a mass transport effect. And then on the right there's no mass transport effect. You can fit this in all the models if you do have a little bit. Now for BLI, the controls are a lot easier. So what you have then is if you have the red column there, that's your ligand. One of the sensors will just go into buffer. So that's just going to be unaddressed. So that then you're going to put that, when you put that into then column 5 there, where you've got the binding partner, the analyte, you could actually see if there's any non-specific binding. So here in red, you can kind of see there's no ligand going on, and there's no interaction with the analyte. And again, in channel 5 as well, we've got spot G there, is got just buffer, and we can look then against what the background of kind of the instrument is like. Alternatively, what you can do if you need lots more concentrations is you can actually run things in parallel. So you could have eight sensors, and then which will be addressed with ligand, and then 8 sensors which don't have any ligand on. But then, of course, you need then double the kind of number of rows of ligand at the same concentration, but you can do that all in parallel. With switch sense, then it's all built in. There's two different colors of nanolever with different sequences. So they'll be different complementary nanolevers. And so you could bind, one of your proteins to, your ligands to one color, and then look at the non-specific interaction on the other color. Okay, so what do you do if you get non-specific interaction? This is really the kind of the killer for lots of these kind of experiments. So you can change the chip. So you can change, so in say CM4 and CM5, CM4 has got slightly less dense matrix of dextran. You could use PEG, it's a different type. You can cross-link with PEG, or you could change the buffer to the conditions and try and get that over, or you can put in additives. So things like Twing are always good, or you can have blocking agents like BSA, EDTA. There's this thing called non-specific binding reducer, which they sell, which is essentially just soluble carboxymethyldextran. And obviously for streptavidin, you could block with biotin or biocytin. So what kind of experiments are we going to do? How many concentrations do we need? Kinetics, about 5 concentrations with a 0 control, do things in duplicate or triplicate. And then you need an adequate concentration range if you're doing affinity measurements, you need to be above and below the KD, so you can go all the way through. So what kind of signal and what kind of kinetics you're going to get depends upon the individual rate constants. So let's just imagine you've got 4 analytes with the same binding constant, but they've got different individual on and off rates. So what's the shape going to look like? Okay, in blue, it's very, very fast on, but it's also very, very fast off. Then as you go down, you can kind of see at this one concentration, you're getting slower observed on rates, but very, very slower off rates. And at the bottom there, you can see that, okay, red is really slow on, but because it's got a slow off, it's actually going to occupy the ligand for a much longer time. As we can kind of see here. So you can imagine here in a screen of small molecules, we've got on the x-axis, we've got log off. On the y-axis, we've got log on. And then the parallel stripes across are all KDs. And so you can see there's a whole buffer you can kind of get. So if we just look at compound 1 and compound 2, compound 1 is kind of slow on, slower off, while compound 2 is very fast on and fast off. So actually compound 1 might be a better one to develop because it's got slower off. So this is the two extremes. You're going to have a steady state where just things are going on and off. You get this kind of equilibrium position where you've got a flat line at the very top after the injection, or you got kinetics where it's basically you can't see that flattening off until you get very high concentrations. So we're going to fit the data. We're going to subtract the reference binding, and then we're going to do a double subtraction. So it's called double referencing by taking away the buffer control. And we can fit this either in the program that the company supply, or we can take the data out as text files and then just fit in Prism. Now for switch sense, we've looked at everything kind of going up and coming down. For switch sense, it's the other way around, because when you get binding, you're going to then get a decrease in the mobility of the nanolever as it's kind of trying to get back up, moving across. And so then when things dissociate, then the speed of the switching then goes up. And you can actually fit things either locally or globally in the program that they supply. Okay, so for equilibrium binding, all you have to do then is just take what that equilibrium response is, fit that against the concentration of your analyte, and then just a simple equation gives you then what is 1 over the dissociation constant, which is called the association constant. Kinetics is a bit more complicated. Okay, so trigger warning, there's going to be some equations. But, okay, so you're going up in concentration. You've got on the left-hand side, sorry, on your right-hand side, we've got a dissociation. Now, that just depends upon the dissociation constant. But when you get association, that observed association depends on both the association rate constant and the dissociation rate constant. Unfortunately, you don't really have to kind of think about the equations a lot because there's lots of fitting, automatic fitting you can do, but there is lots of different models you can put in, which is a danger, obviously. Because the more complicated the model is, the better the fitting might be because you're just putting in more parameters. So better to get good data to get good fitting. Right. So let's think about the dissociation first. Right. It's pretty simple. It's just a little simple exponential dependence then just on the time and then the rate constant. So this is dependent on both those things. And this is the equation here. But it's important to think about the fact that in this equation, you'll see for fitting this first phase, that you have to know what the dissociation constant is. So what if that is wrong? Here's the case, right, which we did a while ago. And if you have a look at the off rate here, or this dissociation phase, it's pretty flat. It's hardly dissociating at all. It's very, very difficult to actually fit because you're not getting really an exponential. You're just really looking at this kind of almost straight line. So the computer will fit it. It will give you a nice error, very low error, but it may be wrong. In this case, it estimated that the binding was like 30 nanomolar. Okay, so ways around that is, if we can't believe actually this is really fitting quite well, fitting dissociation, is we can look at the association phase, and we just measure the observed fitting. There's again a simple exponential for 1-to-1, and then we can fit that against then the concentration of analyte. And for 1-to-1, that should really be a straight line where the gradient is going to give us association rate constant, and then the intercept on the y-axis is going to give us the dissociation rate. And for this case, if we did that, instead of 30 nanomolar, it gives us a KD of 1.8 micromolar, and by ITC is about 2.2. Okay, so how do you make you have a choice of these kind of different evaluation models? Pick the simplest one first. Don't make things complicated. What does it look good? How is it good? You look at the residuals. You can actually visualize things. What's the chi-squared for the fit? And for BLI, then in the data that you get out, it will give you this ratio of equilibrium over R max, and that should be between 10 and 90%. And again, don't invoke a more complicated model just because you actually get better fitting. Okay, so let's just go on to some examples that we've done here in projects. So this is in the Barford lab. They wanted to know how this APCC was activated, and because it's a big, Cullen ligase, it gets activated by CDH1. How does that affect its binding then to its E2 ligase? So obviously, this is, you can see, it's a huge, big complex, and we didn't really want to attach that onto the surface because, well, it may just fall apart, and then what have we got? So then we attached then the small little UBCH10 onto the surface, and then looked at the interaction between the with the APC plus and minus in the CDH1. So easy there with streptavidin, you just get the binding level you want by just increasing more and more and more until you get to a level you think, okay, that's good. Unfortunately, when I put the APC in, it was horrible. It bound onto the surface, and it's like, so there's no protein, in this channel, there was no protein there. It was just like, well, what am I going to do? This is, there was referees kind of waiting for these results that, so the paper was being delayed by this. So then I think, okay, well, let's go and try and reduce it. So I just went through kind of different kind of things that will reduce it, tried them in all combination until then I got conditions here, and this was with tween and salt. So tween and BSA and NBS then down the bottom there to try and, or I think it was just with tween and BSA actually, to get us down to a nice low level of non-specific binding. So the experiments themselves, very easy to set up. Just as computer driven, just say, oh, I want to do this experiment. Okay, well, how long do you want to interact with? What do you want to regenerate with? What concentrations are you going to put in? It'll ask you, and then it will tell you, oh, put all these these things in this rack in these positions here. You hit go, it'll give you a kind of results, and then you can actually then process the data on the computer itself, and then it just fit the data. Oops. And then in this case, what I did was I took the data out and fitted it in Prism, and I could fit the on and the off rates. I could actually, because we're getting to equilibrium as well, then I could actually fit the equilibrium binding constant, and also, just for a bit of fun, fit the observed rate constant as well against concentration. And actually all of these then fitted very, very nicely. And the equilibrium binding was essentially the same as the kinetic, the binding by derived from the kinetics. So that was nice. Okay, for switch, so for SPR, you can do ligand screening as well. And this is some data from Javier. So the problem is, of course, if you're screening a library, you're going to get very, very small little signals. So everything has to be very much controlled. And so you need a sensitive biocore. And I mean, you can do free proteins up in parallel, and also you can screen in 96 or 4-well plates. One big problem is that lots of these compounds are actually in DMSO, and they're maybe kind of stored in a fridge or on a shelf or whatever. DMSO is very, very hydroscopic. So if these have been sitting around for a long time, the actual DMSO isn't 100% DMSO anymore. It could be like up to 20% water. So you need to correct for that. And luckily in the program, there is a correction kind of wizard where you put in different concentrations of DMSO so that it can kind of correct for those kind of non-specific changes in refractive index. So the kind of data you get then, if you're doing a screening, is you can see kind of a whole forest of points. So it's always good to have positive and negative, so that then so, things that don't bind and things that you do that does bind. So you can kind of set a kind of a window, and you can kind of rank what's binding. And you can see in the middle there was lots of hits, and then above at the top there you can see there's 3 ones that were actually going way above, kind of super binders, and that's going to tell you these aren't good either, because these are probably just maybe aggregated compounds that are just clumping, because you have an expectation of what the kind of the one binding level would be. Okay, so for BLI in action, we come back onto COVID. This was a study to try and make a more stable trimer of the spike protein for vaccine development. And so we got biosensors. In this case, we got the ACE2 that's attached to an FC. And then it could be bound onto protein A sensors. So if we're going from the left, then we see we've got baseline, bind on the ACE2 FC, put it into then the column with the spike protein. We get lovely kind of binding, and then into buffer for the dissociation. And then at the end, then to get the reference, what I did was then I could disrupt the FC or the ACE FC protein A interaction by just then putting it into regeneration buffer, and then use the same sensors again through the analyte again, just to see what the background binding was, and it wasn't anything at all, so it was good. And of course, so what you get is, this is the corrected data. You can see there's little jumps there at the end. We can correct for that. That's just because it's going into different size plates. And so we can correct for that, fit the data. And in this case, then we could fit it very nicely. And we could kind of see that there was 2 phases of fast and slow on, but more importantly, a slow off. So it was estimated then to be like 3.6 per 10 to the minus 5. So you can work out what the half times that would be, and that's quite a long time. So once it binds, it won't come off for a very long time. Right, for switch sense, you start off with a nanolever that's bare. You flow across your ligand that comes on, and you measure the interaction. And then you can then disrupt then the nanolever by then just washing, and then get rid of it and part the whole cycle again. So here's an example here from the Passmore group. And so this was James. And so we have this construct, a bound RNA, and so this is YTH. And it seemed to be from from bioinformatics that there's an upstream region that seemed to be quite conserved. Why it was conserved? Well, it was interesting. So when he put it into a fluorescence polarization assay, the domain, for the domain by itself, found here, kind of in the high, in the micromolar nanomolar, high nanomolar range. But then if you had this upstream unfolded region, well, the binding looked really, really tight. So we made this DNA RNA ligand here. So you can see that the RNA has to be 5', and then it goes into DNA at 3', because then that's going to be complementary to the nanolever. So we flow that on. So here's the association. You can see the association between the domain itself and with and without the unfolded region. Look pretty similar kind of range. But when it came to look at the dissociation, you can see it's very much different. Look at the time frame. Dissociation is about 5 minutes here. When you've got the unfolded region, it's, that's up to 360 minutes. So much slower. So we can see then that this unfolded domain is really important then to kind of slow the kind of dissociation off. Further information, look at the websites of the different manufacturers, go onto our website internally, have a look. Thanks for your attention. Thanks. Any questions?